Okay, so this is a video on protein synthesis, which is basically the making of proteins inside your cell based on your DNA. And it all starts by making a molecule of RNA. And the first one of these molecules is mRNA, which stands for messenger RNA. And this happens in a process called transcription, which happens inside the cell nucleus. So here we have the cell nucleus, here's the nuclear envelope, and here's the nuclear pore there, which is an opening on the nuclear envelope. And this process starts in a similar way to DNA replication. You have the DNA double helix here, and a little enzyme called DNA helicase will come in and again break the hydrogen bonds between the complementary base pairs, and so separating the two DNA strands. Then, in a similar way to DNA replication, the enzyme polymerase will come in, and this time it's not DNA polymerase, it's RNA polymerase, because we're making a strand of mRNA. And this will basically come in and join the complementary free nucleotides within the nucleus to the complementary bases on the DNA strand. So this will form a molecule of mRNA. This molecule will then leave the nucleus via the nuclear pore. But before this happens, it's important to understand that in RNA, they don't have the base thymine. Instead, they have the base uracil. So here's a kind of little diagram to represent that. So for every adenine on the DNA strand, instead of having a thymine, you'll have a uracil. Guanine and cytosine are the same. For every thymine, you will have an adenine, and it will go along like this. But for every adenine, you will not have a thymine, you will have a uracil. And that's just one of the things that makes RNA different from DNA. Also, 3 of the bases on an mRNA strand, they're called a codon. So it's not called a triplet code anymore, it's called a codon. And also, after the mRNA has been made, the molecule is spliced, meaning the introns are taken out of the molecule, making the molecule considerably smaller, and it's just left with the exons, which will code for amino acids. Okay, so we need to remove this. No laughing. And the next process is called translation, which is the next part in protein synthesis. So this involves making a polypeptide chain from the codons of the mRNA strand. And this happens outside of the nucleus, and it uses these ribosomes, which, as you know, are the site of protein synthesis in the cell. And this also uses another RNA molecule called a tRNA molecule. And these have anticodons on them, which are complementary to the codons on the mRNA strand. And what the ribosome does is it's kind of like a zip, and the zip will go up the mRNA strand and attach the complementary tRNA molecules onto it. On the other side of the tRNA molecule, there is a amino acid binding site, which is where it holds its complementary amino acid. When 2 tRNA molecules are attached to the complementary mRNA strand, the amino acids on the end of them will form a peptide bond. This is the start of the making of the polypeptide strand. Then, as more tRNA molecules come on, others will leave. So the ones that have already got their amino acids joined together, they will leave. and they will go off to find more of their complementary amino acids so that they can form more polypeptides. And this will continue to happen. They'll continue to form peptide bonds until the ribosome reaches a stop codon on the mRNA strand. When it's reached the stop codon, it will not code for any more amino acids, which will basically stop the polypeptide chain from being made. This polypeptide chain will go on to form the proteins, and these proteins are what causes the chemical reactions in your body. So it's basically all the characteristics are determined by the base pairs in your DNA, because the base pairs in your DNA code for the mRNA, which code for the tRNA, which have the amino acids, and these are what form the proteins. So that's about it for protein synthesis, but in the next video I'll talk about mutations, which is where you have a change in the base sequence of the DNA, which obviously leads to a change in the mRNA, and so you will form a different protein.